Good afternoon, everyone. Welcome to this session of physiology. We are going to be covering today some important concepts that have to do with the genetic control of the cell functions. Let's proceed to take a look at the learning objectives that we have for today. We are going to be covering certain concepts that are essential in genetics, like chromosome, genes, DNA, RNA, codon, anticodon, protein synthesis, gene expression, chromatin, so we can understand how DNA and RNA and the genetic code help us to understand different inheritable conditions, genetic diseases that may appear due to mutations, and also due to a combination of genetic predisposition and some environmental factors. We are going to be understanding some key genetic terms like genotype, phenotype, alleles, dominant, recessive, homozygous, heterozygous, etc. And applying the principles of Mendelian inheritance so we can predict what may happen, for example, in families. Okay, what is the proportion of people who may inherit a mutation or may manifest a disease. We're going to be studying the different types of mutations and, of course, predict what may happen as a result of these mutations. Okay, besides this, we are going to be talking about the role of epigenetics in disease, trying to understand how some people may have a DNA with no mutations at all, the normal sequence of bases in the DNA, RNA, etc., and they may have some problems because of what we call epigenetic factors. Okay, epigenetics is a science that is fairly new. Okay, the term epi or the prefix epi means something that is beyond or above, over the genetic composition of the person. So let's proceed. Let's start analyzing some of these concepts. Okay, in the first slide here we have what we call the human karyotype. In this case, it's a female karyotype because, as you can see there, there are a couple of chromosomes up here, chromosomes X. I was saying 22, no, this is the 23rd pair. So we have here the two X chromosomes. If that tells us that this is a female individual, if instead of a female we had a male karyotype here, there would be one of these X chromosomes and a tiny one that is the Y chromosome. Okay, the human karyotype, okay, any human karyotype will contain 22 pairs of autosomes, which are the chromosomes that are not sex chromosomes. And then there are the sex chromosomes, which are known as allotypes or simply sex chromosomes. Okay, we have 46 chromosomes in total. We inherit half of them from our mothers and the other half from our fathers. Okay, the chromosomes are these long thread-like structures that are made of DNA and proteins. Okay, and they contain several genes. Okay, we assume, we have calculated that humans have 22,000 genes. Okay, so it's a huge amount of information that can be used to make different proteins and also to control the functions of the cells. Let's delve into one of the chromosomes. Let's take a look at the human chromosome 4. We chose this randomly. We could have chosen any of the chromosomes to show the same idea. Okay, each chromosome is going to be made of certain amount of DNA that is coiled around some proteins that are called histones that allow the cells to pack very tightly the DNA. Okay, the cells need to pack the DNA very tightly in the nucleus so the DNA is protected, and also so we are not constantly transcribing the DNA into RNA and making proteins all the time. We make sure that the cells only synthesize, make the proteins that they are going to use at a certain point, the proteins that they need at a certain point. Okay, the DNA has been called the molecule of life. It's like a library. Okay, all the genome of the person or the animals or the plants is like a library that contains instructions of how to make proteins that the organism will use. Okay, each of our cells contain exactly the same DNA, 46 chromosomes in 23 pairs. And this is true for all cells except for the red blood cells that are not true cells. They are simply little sacs that contain hemoglobin and transport oxygen. Okay, here you have some information about the human DNA. Okay, if you uncoil the DNA, it would measure like 2 meters. Okay, there are around 3 billion DNA base pairs per set of chromosomes, and each of them contain these 4 bases: A, T, G, and C: adenine, thymine, guanine, and cytosine. Okay, and there are, as we said before, approximately between 20,000 and 25,000 genes, okay, that code for proteins. Now let's take a look at the composition of these chromosome 4. Notice that the chromosomes can be subdivided into genes. Okay, there are several genes. Each of them will code for a different protein. And notice that there are some diseases associated to mutations and associated to different problems in each of these parts of the chromosome. Okay, a gene is simply the piece of the chromosome that contains specific functions to make a specific protein, and every gene will be located in what we call a locus. A locus could be like the addresses where the gene is located. Okay, the locus is like the location of a specific gene within a chromosome. So this DNA, okay, deoxyribonucleic acid, carries the instructions, okay, for cell growth, protein synthesis, development, reproduction, okay, and is composed of 2 strands, as you see here, that coil around each other to form a double helix structure. Okay, and the primary elements of this DNA are the nucleotides that we mentioned before: adenine, thymine, cytosine, guanine. And they are, if you see here the double helix, you see the bases inside this double helix. Okay, there is a hydrogen bond, or 3 hydrogen bonds between cytosine and guanine, and 2 hydrogen bonds between thymine and adenine. Okay, always you're gonna find a T paired with an A with a T, and a C with a G, or a G with a C. Okay, that is also always going to be the case in the DNA. Okay, and there is a backbone. These blue lines are the backbones that are made of sugar and phosphate, and that sugar is deoxyribose. Okay, and there are phosphate groups as well. Let's take a closer look at that double helix. You can see here the phosphate and sugar backbone, the blue structures here, and the bases in the center: guanine with cytosine, cytosine with guanine, thymine with adenine, thymine with adenine, etc. Always match in that way. And here you can see the 2 hydrogen bonds, okay, between adenine and thymine, and the 3 hydrogen bonds between cytosine and guanine. Now, there is a law, let's call it a law in biology, okay, that we call the central dogma of molecular biology, which is described as the unifying theme of molecular biology. And this simply expresses or represents how the genetic information flows in a cell. Okay, we start here in this gene in any chromosome, could be any gene in any chromosome. And this gene, for example, when the cell divides, okay, we first replicate all of our DNA to make an exact copy that will be transmitted from the parent cell to the 2 daughter cells. Okay, now the information, the genetic information, will flow from the DNA to RNA in a process that we know as transcription. This is a process that occurs in the nucleus. Okay, a segment of the DNA molecule unzips and serves as a template to create a corresponding, a matching piece of messenger RNA. that process occurs in the nucleus, the transcription. Okay, now the messenger RNA, okay, will carry the message from the DNA to ribosomes that are outside the nucleus. They may be present in the rough endoplasmic reticulum, or they may be present in the cytoplasm as free ribosomes. And these ribosomes are gonna read the information from this messenger RNA to make proteins. So the messenger RNA could be considered a temporary copy of a gene that contains information to make a polypeptide or a protein. Okay, the process that occurs in the ribosomes, rough endoplasmic reticulum, or free ribosomes is called translation. Okay, this is the process that makes the protein using the information from the messenger RNA. That is, as we said, a transcription from the information in the DNA. Now, these polypeptides are a chain of amino acids that later have to undergo certain transformation. Okay, the proteins that we have in the body are not simply a chain of amino acids. They acquire a tridimensional structure, can be globular, like round, or can be forming helices, okay, forming fibers, and there are different types of proteins, okay, antibodies, keratin, hemoglobin, collagen, okay, that each of them will have a different function, okay. Examples of proteins also are the channels, the receptors that we have in the cell membranes. Okay, so that is how the way the information flows from DNA to RNA to protein, and that is the central dogma of molecular biology. This is what normally occurs in our eukaryotic cells. And we have to also cover, okay, a concept that we are gonna be referring to, okay, very frequently, and is the process of gene expression. What is gene expression? Well, this is a process, okay, by which the DNA, okay, the instructions that we have in the DNA, in these genes, is converted into a functional product or protein. And there are two steps, transcription and translation. Okay, notice, if we go back here, that replication is not part of the gene expression. Replication is what occurs when the cells are going to divide. Okay, transcription and translation are the two steps of gene expression. Okay, the sequence of DNA nucleotides is transcribed into a complementary sequence of RNA in the transcription, and then this RNA is processed. Okay, we are gonna see part of this processing in the next slide. Okay, where the RNA is translated into a sequence of proteins by the ribosomes. So let's take a look at this processing that occurs, okay, in the genes. Okay, before explaining that, let's take a look at the gene structure. I found this diagram online, and I had to choose this among several others that had very, very complicated information. Okay, here we have the basic information that we need to know or to get to understand gene structure and how the gene expression is controlled or is regulated. Okay, what we have here? Okay, we have here certain components of a gene. Notice that there is a region in a gene that we call the promoter. Okay, and then we have some structures here that we call exons. And in between the exons, we have other regions that we call introns. Okay, so there is a region of a gene that is transcribed, and there are regions of genes that are not transcribed. Okay, the promoter is one of those regions, but besides the promoter, there are other regions that also regulate gene expression. For example, the regulatory sequences, or a region called the terminator. So, for example, if you want to start producing the transcription of this gene, okay, what you normally do is bind a transcription factor, which is a molecule, okay, to the promoter, and that will initiate the transcription of this region of the DNA that is the transcribed. Okay, the promoter is the starting point. Okay, it's a DNA sequence that is recognized by the enzyme RNA polymerase. Okay, and the enzyme binds there to begin the transcription. It's like the starting point. The coding region is the part that is transcribed. Okay, the terminator is the end point of a gene. Okay, that terminator sequence tells the enzyme RNA polymerase to stop the transcription at that point. And there are some regulatory sequences that increase or decrease the amount of RNA that we are going to make from a single gene. Okay, there are two ways that we can control the amount of protein that we make. One of them is by making more or making less of this messenger RNA. Now notice that we transcribed all this region of the gene, okay, but at the end, in the messenger RNA, we are going to have only the exons and not the introns, or the introns are removed. Okay, and we have the final messenger RNA structure, which are only the exons without the introns. Okay, let's look at more details here. Okay, here we have these other structures that we mentioned before. There is a promoter. Okay, this is the transcribed region, and then we have the terminator that didn't appear in the previous slide. Okay, we have different steps in the transcription of a gene. The first step is initiation. Notice the RNA polymerase binding to the promoter and starting to open up, separating the two strands of the DNA and making a chain of messenger RNA. Okay, that will occur in the process that we know as elongation. And then we have the termination, when the RNA polymerase gets in contact with the terminator. Okay, we have at the end of the process the completed RNA transcript, the messenger RNA, and the RNA polymerase detaches from the DNA and the process stops. Now this is regulated, as I said before, by simply limiting the amount of messenger RNA that is produced. If we need to make more protein, we are going to make more messenger RNA. If we don't need too much of this protein, we simply produce less of the messenger RNA. Okay, there are a number of regulatory transcription factors that will activate or inhibit the transcription. Okay, the chromatin, which is the way in which the DNA is packed in the nucleus, okay, can be more dense or more relaxed. If it's dense, something that we know as euchromatin, okay, there is gonna be no transcription at all. Let me write this down here. Okay, the chromatin can be found as euchromatin. In that case, there is going to be no transcription because this is very dense. And then we have the heterochromatin, which is more relaxed. Okay, and in that case, okay, we have transcription. Okay, the DNA is like exposed to the action of the RNA, to the enzyme that makes RNA from DNA. Now, another way of regulating gene expression is by some post-transcriptional events, okay, that regulate the translation process, not the transcription. That means we can regulate at the level of the transcription and at the level of the translation. Okay, there are many different proteins. There are some small segments or pieces of RNA that are called microRNA that silence the translation of messenger RNA, and there are many other processes, okay, that also participate in increasing or decreasing the amount of messenger RNA that is translated. Okay, here we are going to see some of these post-transcriptional modifications. okay, in which the immature messenger RNA is converted into a mature messenger RNA. There are different processes like what we call capping, is the addition of chemicals, a 5' cap at the end of the messenger RNA, and this is important for ribosome recognition. Ribosome will know that they have to bind here and start reading, okay, when they recognize this 5' cap. Also, the addition of many adenines here, okay, there is a very long tail of adenines, poly A tail, that is added to the 3' end of the messenger RNA. Notice that there can be between 150 and 250 adenines there. That is important to prevent the degradation of the RNA as it travels from the nucleus into the cytoplasm and until it reaches the ribosomes. Okay, splicing is another process, okay, that allows the removal of the introns and joining the exons together. Okay, there can be different types of splicing. Okay, and if we, for example, study, okay, the production of antibodies, we can see that there can be many alternative ways of splicing the RNA, so we can obtain different proteins that are specific, okay, to fight different types of bacteria, viruses, etc. Now, the last step here is translation, okay? The messenger RNA carries the transcribed message from the DNA to the ribosomes, okay? Ribosomes read, quote unquote, read this information, okay? The transfer RNA will start reading 3 bases at a time, okay? And so I had to make a little stop here to correct a mistake that was there in the slide. I already fixed it. And we were talking about translation, okay? And in this case, the information is read by the ribosome, okay? The ribosomes are gonna read 3 bases at a time, okay? They are gonna read what we call the codons that are in the messenger RNA or on the messenger RNA. Okay, we are going to be seeing how it works when we move to the properties of the genetic code. Okay, then once the ribosome reads the codon on the messenger RNA, okay, we are going to have a molecule of transfer RNA that has a complementary anti-codon, okay, complementary to the codon on the messenger RNA, and that transfer RNA will bring a specific amino acid, okay, attached to it. We are gonna see in the next slide how it works. And this is the example of a codon, GGU. Notice that here we have uracil that tells us that this is RNA and not DNA. Okay, GGU, this codon will code or will bring a transfer RNA that has the amino acid glycine attached to it. Okay, all these things are better understood when we look at videos, when we look at animations. Okay, you have many of these great animations on osmosis, and also you can find videos online. It's very hard for us to have that degree of abstraction and imagine how this works simply by reading something that is a summary of the summary in a PowerPoint. Okay, now the ribosome reads this codon and then moves to the next codon. Another transfer RNA will bring another amino acid. The ribosome will form peptide bonds between these 2 amino acids, and this process is going to be repeated over and over, and we are going to have a long chain of amino acids that later is going to be acquire certain tridimensional structure. Okay, so we are going to see the genetic code. We are going to see what are the codons. We are going to see what is a start codon. We are going to see what are the stop codons and what codons code for different amino acids. Okay, we are going to see in the next slide that there are 20 amino acids that form our proteins. But when we look at the genetic code, there are 64 potential combinations of codons. Okay, so we don't make 64 amino acids, only 20, because of these 64, okay, well, 4 of them are either a start codon and or stop codons. So the other 60, okay, can code for more than one amino acid. So we have 60. If we don't count the start and stop codons, we have 60 potential combinations to make the 20 amino acids. More than one codon can code for the same amino acid. So let's look here at the genetic code properties. Okay, the genetic code, as you can see here, is made of triplets. It's a triplet code, comprises codons and sequences, which are sequences of 3 nucleotides. Each codon corresponds to a specific amino acid, or can be start or stop signals. For example, if you analyze this genetic code, okay, we can have in this, here on the left, we can look at the first letter. The second letter is here, and the third letter is on the right side. So the codon uracil, 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 and the codon uracil, uracil, cytosine, they will both code for the amino acid phenylalanine. Okay, this is very important to analyze when we start studying the mutations that may occur in the human genome. Okay, in these mutations, there can be a substitution of one of these letters for another. Okay, so notice that, for example, if someone has to incorporate phenylalanine in a protein, and the original code has the codon UUU, and there is a mutation that substitutes uracil for cytosine, then nothing is going to happen. We still will have the same amino acid there. Now, if instead of changing uracil for cytosine, we add adenine there, then we are not going to produce phenylalanine. What we are going to produce is leucine. Okay, now if the original code is UUA and we substitute adenine by guanine, then nothing is going to happen. Okay, that's how we interpret that. That's how we understand that we have 64 potential or combinations, but we only produce 20 amino acids, the ones that we have there. Okay, there you have the start or the starting codon, start codon, that is AUG. There is a mnemonic, inaugurate, to remember that. And there are 3 stop codons, UAA, UAG, UGA. There are also mnemonics to remember that. Those are you are away, you are gone, you go away. Okay, these are the 3 mnemonics that we typically use to remember which codons are the stop codons. Now, besides having this property of being a triplet code, the genetic code is universal. That means all species, eukaryotes and prokaryotes, have the same code. Okay, the genetic code is degenerate or redundant. The word degenerate doesn't sound very well, okay, but it's one of the properties of the genetic code. This simply means that more than one codon can code for the same amino acid, as the example we mentioned there. For example, UCU, UCC, UCA, UCG, the 4 of them code for serine. Another property is the genetic code is unambiguous and non-overlapping. Okay, non-overlapping means the ribosome is going to read the letters, the bases, 3 at a time. Okay, 3, then 3, then 3, then 3. Okay, it's not going to read 2 and then 3 and then 1 and then 3. It's simply 3 at a time. Okay, and unambiguous means that, for example, from CUU, we are going to make, or we are going to incorporate, not make, incorporate to the polypeptide chain the amino acid leucine and only leucine. Okay, when the ribosome reads CEU, okay, the transfer RNA will bring leucine, it won't bring proline or serine or any other. Okay, so those are the properties. Okay, the presence of start and stop signals, okay, is another of the properties, okay, that is characteristic of this genetic code. And here we have another picture representing the genetic code, just if you want to have it, okay, by itself. We have here the stop codons, okay, the start codon, okay, we can write here the mnemonics if you want. For the start codon, inaugurate. Inaugurate. These are things that we in medicine have to use constantly, the mnemonics. Okay, in medicine we have to remember lots of information in a very short time. So we need to use mnemonics to make sure that we remember all those things. The three stop codons are U, R away, which is UAA. Then we have the U argon, or UAG. And then we have the U go away. U go away. That is the UGA. That's not hard to remember. I saw this mnemonic once and I still remember. Okay, so that is all the information that we need to get about this genetic code, about the regulation of gene expression, steps of gene expression, about the concepts of DNA, RNA, chromatin, okay, and about all the details that we need to know, okay, for understanding later genetic diseases, genetic inheritance patterns, etc. Okay, we are going to move now to try to understand some of the most common mutations that may occur to this DNA. Okay, mutations are changes in the DNA sequence, in the normal DNA sequence that makes up a gene. And there are many different mechanisms that may result in a mutation. And there can be several consequences for these mutations. Sometimes the consequences are not important. Sometimes they can be very, very bad, can lead to cell death, cancers, etc. Okay, they are defined as a permanent change in the nucleotide sequence. These mutations may arise during replication or recombination of different genes. And there can be, for example, mutations that are classified as substitution, also known as point mutation. There is a substitution of one base by another base, as we saw previously when we were explaining how the genetic code must be seen. Sometimes a substitution doesn't have any consequences, but sometimes it can have very bad consequences. There can be also deletion or insertion of a base. Okay, most of the mutations are totally harmless, okay, but in some cases they can lead to very bad consequences. And there are several mechanisms that the cells have to repair the damaged DNA. There are enzymes that proofread the DNA and repair the DNA if there is a mistake. And if the mistake can't be repaired, the cells normally will stop. Okay, there are mechanisms that stop the cell division, so we don't create new cells that carry the mutation to new generations. Let's take a look at some of these examples. Okay, let's start with base substitution. Okay, this is the most common type of mutation, point mutation. It's a single base substitution. Okay, there are characteristic diseases, for example, sickle cell disease, in which there is a substitution or leads to a substitution of one amino acid for another. Okay, glutamic for valine. Okay, mutations can be of different types, silent, missense, or nonsense. A silent mutation will be one in which the substitution occurs generally at the third position of the codon. Remember the examples we saw. If we change this base here, typically we don't have a change of amino acid. Okay, instead of U, we place there a C, we are going to have the same amino acid. Okay, or if we have a G instead of an A, we're going to have the same amino acid. Okay, in this case we can have a different, it doesn't matter what base you use in the third position, we always will have proline. So this silent mutation, okay, generates what we call a synonymous codon, a codon that codes for the same amino acid and will have no phenotypic effects. So it's not going to manifest with any trait, okay, at all. Now then we have other two types of point mutations, the missense and the nonsense. Okay, a missense mutation is one that leads to the codon that codes for a different amino acid. Okay, now the phenotype effect of this missense mutation will depend on the properties of the new amino acid. Okay, some amino acids have similar chemical properties. They may be of the same type. For example, both may be hydrophilic or hydrophobic, or they may be more alkaline or neutral or acidic. So if we substitute one amino acid for another that is similar, okay, the changes are going to be mild, maybe non-noticeable, and we call that type of mutation a conservative mutation. Now, if we have a totally different amino acid, let's say instead of a hydrophobic amino acid, a hydrophilic amino acid, now this will have a very important repercussion in the three-dimensional structures of the protein, okay, and that will manifest with a genetic disease, a protein that may be totally afunctional. Okay, these are called non-conservative missense mutations. And the nonsense are the ones that lead to the formation of a non-functional short or truncated protein, because we simply have a substitution, okay, of a base, and we have and this substitution results in a stop codon. So let's imagine that we normally have a protein that is 1000 amino acids long. And just to use one example, let's say that the amino acid 500 is UGC. The codon is UGC that codes for cysteine. If we have a substitution and instead of a C, we place there an A, now when the RNA polymerase reaches this point, it will find UGA, okay, and that will stop the protein, RNA polymerase, no. When the ribosome gets to this point in the messenger RNA, instead of continuing adding cysteine and the rest of the amino acids, it will simply stop and will release the protein, and it's going to be a short afunctional or non-functional protein. That's why we call it nonsense. If you need a mnemonic for that, the one that I used when I was preparing for the boards for the USMLE was non-stop the music. Non-stop, non-stop. So I remember that the nonsense mutation is the one that produces a stop codon, leading to the formation or the synthesis of a short non-functional protein. So these are the types of mutations regarding base substitution. We are going to see now what happens with deletions and insertions. Okay, these deletions and insertions typically always lead to a dysfunctional protein, and we are going to see why. Okay, here we have examples of what can happen when we have deletions and insertions. This type of mutations will lead to what we call a frame shift in the reading sequence. Okay, here we have an example of the original DNA code. We have CAT, CAT, CAT. Notice that is a polypeptide chain that is made of many histidine, histidine, histidine amino acids together. Is CAT, 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 CAT. Okay, but if we have the insertion of a base here, now instead of CAT, 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 etc., we have an A where the C should be. So now, instead of CAT, okay, the triplet reads ACA, and that doesn't code for histidine, that codes for threonine. Okay, notice here the CAT, the original one. Okay, we have the insertion of this A here. So now what is next? What is the next sequence? Instead of CAT is TCA, and this is gonna code for serine, and then we continue TCA, TCA, serine, serine, serine. So produces a totally different protein. Okay, the same may occur if there is a deletion. Instead of adding a new amino acid, we remove one amino acid. Let's say we remove this C. So when the ribosome is reading this chain, it's gonna read cat, cat, cat. It's gonna place histidine, histidine, histidine. But now instead of reading cat, it's gonna read ATC. Okay, ATC and then ATC, ATC, ATC. Okay, and if you want to know what amino acid goes there, we look at the first letter here, A. TC, we mean the original sequence in the DNA. Okay, in the RNA we don't have a thymine. Okay, what we have is a U. Okay, uracil instead of a thymine. Okay, so that is gonna be the consequence of this deletion in the DNA sequence. There are more, okay, that we haven't represented here in diagrams, okay. Besides these insertions and deletions, there can be duplications when we copy a segment of the DNA more than once. There can be inversions. The original DNA sequence may be reversed within the chromosome. There can be translocations when we have a segment of DNA, a gene, for example, moving from one chromosome to another. Okay, we are gonna see when we study pathophysiology, when we go to physiology 2, we are gonna see some examples of these translocations and what happens as a result of different DNA mutations. Okay, here we have some examples. This is just an illustration for you to see the different sequences in the DNA, messenger RNA. Okay, it's a normal chain that leads to phenylalanine, tyrosine, glutamine, glutamine. Okay, notice that here we have a substitution. Instead of GAA, we have AAA that codes for lysine. Okay, here we have an insertion, here we have a deletion. It's exactly the same examples, but here you have some everything together in one slide. Well, besides the genetic mechanisms, okay, we have to talk about epigenetic mechanisms, because not everything is in the genes. Okay, the genes interact with their environment. Okay, there are many different things that get in contact with our genetic material, sometimes viruses, sometimes toxins. Okay, and this may produce some temporary or permanent alterations in the DNA, okay, that either may silence the DNA or may activate the DNA. Okay, and this is sometimes useful, for example, when during the period of embryonic development, okay, at the beginning, all the cells are capable of doing everything, okay, but then when the cell start specializing, okay, we don't want cells, for example, of the pancreas start to make collagen, or we don't want cells of the skin to make insulin. We don't want cells in the retina to make sweat, for example. You don't want cells to produce something that they shouldn't be doing. So we need to silence some genes, we need to activate other genes depending on where the cells are located. Okay, and these mechanisms that control the gene expression are called epigenetic mechanisms. Okay, we have here some examples. Okay, one of them can be histone acetylation. And this is a process that we add an acetyl group to histones, these proteins around which the DNA coils. Okay, when we acetylate these histones, that will remove positive charges and decrease the attraction of the proteins, the histones, to DNA. So that will uncoil, okay, loosen the DNA and make it more available for transcription. Okay, that activates the DNA when the histones are acetylated. If they are hypoacetylated, the contrary happens. Okay, that silences the DNA. Okay, another mechanism is histone or DNA methylation. That is the addition of a methyl group, okay, and that will render a silent DNA. Okay, methylation starts with M, acetylation with A. You can use that as a mnemonic. You can use A for active, activates, M for missing, or you can also say, for example, mute is another mnemonic that I've heard as well. Silences the DNA. Okay, and for example, the X chromosome typically is silenced by a dense methylation. If there is an abnormal methylation, this can lead to silencing and inactivation of genes that we call tumor suppressor genes that typically are stopping the cell growth and division when there is a mutation that can lead to a cancer. If we silence these tumor suppressor genes, of course, the cell is going to grow without control. And that is basically the definition of a cancer. Here we have an example that illustrates very well the concepts of Mendelian inheritance. And here we have two different things. On the left side, we have two parents, one here and the other here. Notice that they have the same genotype, the same genetic composition. Okay, they inherited from their parents two alleles for the color of the eyes. One that has a capital B is a dominant allele, while the other one with the lowercase b, that is the one that codes for the blue eyes, okay, is a recessive allele. So these two persons have the same genotype. Okay, notice that their phenotype is brown eyes. And these here are the possibilities, okay, that they have with each pregnancy, in each of the offspring, of having a different phenotype and a different genotype. For example, 25% probabilities of having one kid in each pregnancy, remember, that has the two alleles that are dominant for brown eyes. Okay, they have 50% probabilities, this one and this one, of having a kid in each pregnancy. Okay, that is heterozygous, so that means has a dominant and a recessive allele. These two here are the same. And 25% probabilities of having a kid, okay, in each pregnancy that is homozygous recessive. So it will have the two alleles that this person will inherit are the two recessive alleles, for ones for blue eyes. Now notice that was talking about the genotype. If we talk about phenotype, notice that there is a 75% probability of having a kid with brown eyes in each pregnancy, and only 25% probabilities of having a kid with blue eyes. Because this blue eyes allele is recessive. That means the person needs to inherit both alleles from both parents in order to have the blue eyes. Okay, that is something that is very characteristic for recessive conditions. In the case of dominant conditions, the inheritance of only one allele is enough, okay, to express the phenotype. Okay, in the next example, on the right side, we have exactly the same. But now we have a parent that has brown eyes and another that has blue eyes. Notice the genotype of these parents. This one here is heterozygous, and this one, the blue-eyed parent, is homozygous recessive. So we could say that, let's imagine that this person here, this person who is heterozygous, okay, is this one here and marries or has kids with someone who is homozygous recessive. These are the probabilities. Okay, genotypically speaking, the genotype, notice that we have 50% heterozygous and 50% homozygous. Phenotypically speaking, the trait that manifests, 50% will have brown eyes because they have the dominant, okay, variant, and 50% will have blue eyes because they are homozygous recessive. So, in a nutshell, for a recessive allele to express as a phenotype physically, outside, measurable, okay, the person needs to inherit both alleles from both parents. Okay, dominant alleles will always manifest. Okay, these are the basics of the Mendelian genetic inheritance. Okay, to understand this, it's important to study the concepts. What is a gene? What is allele? What is a dominant allele? What is a recessive allele? What is phenotype? What is genotype? Okay, we have these concepts in the notes of the PowerPoint, so you can read them there or simply look for them in any reliable source in the book or any reliable source online. Now let's talk about the most important types, the most common types of inheritance that we will see around. Okay, here we have the autosomal dominant inheritance. This, as we said before, when a trait is inherited in an autosomal dominant pattern, an affected individual has 50% chance of passing the trait to each child regardless of the child's sex. Okay, this is because the trait is linked, okay, to one of the alleles in the autosomes, in the 22 autosomes, not in the sex chromosomes. And it's dominant. One copy only is enough for the trait to be expressed. There are examples of these conditions like Huntington's disease, okay, and when we see a family tree, when we see a pedigree, okay, we are gonna see that these conditions appear in every generation. So everybody will have family history of the disease. Now, these conditions, that means that there is no carrier state. Okay, if someone inherits a mutation, the mutated allele, they will manifest the phenotype. Okay, another name for this autosomal dominant inheritance is vertical inheritance, stating that all generations are affected. And, well, it's going to affect equally males and females, but the phenotype, okay, will have differences. Maybe two kids in the same family inherit the mutation, but they don't manifest the disease in the same way because the conditions, okay, these conditions have a variable penetrance and expressivity. Okay, penetrance means what is the proportion of individuals that have a particular genotype that show the phenotype that is expected. Okay, if every individual has symptoms, we can say that the disease has complete penetrance or 100% penetrance. If only 70% of the people that inherit the mutation have manifestations, we say that the disease has 70% penetrance. Expressivity means or refers to the degree or the severity of the disease, the intensity of the phenotype, okay, in those that have the condition. Okay, expressivity varies, okay, can be variable or unvarying. Okay, variable expressivity means the symptoms vary in intensity among people. Okay, for example, a disease that affects the skin pigmentation. Okay, some people may have smaller patches or patches of very light skin, while another person may have larger patches of light skin or maybe not so light. Okay, both express the trait, but the severity or the intensity varies. Okay, this expressivity, okay, can also be described as unvarying or constant. In that case, there is no variation. Okay, everybody will manifest with the same severity. And well, autosomal dominant inheritance typically affects or manifests with disorders that affect structural proteins like collagen. Okay, we are going to see the difference between autosomal dominant inheritance and recessive. Here we have the pedigree. Notice that every generation is affected, and the condition may be transmitted from any parent to either males or females. Okay, here we have a male transmitting to male and female. Here we have another male transmitting to a male, female, female. Okay, and now let's move to the autosomal recessive conditions. Okay, in this case, remember, 2 copies are required. The person needs to inherit 2 copies from both parents, or 1 from each parent, to express the phenotype. If someone is affected, if someone inherits only 1 mutation, okay, these people are not going to manifest the disease. Okay, they are going to be carriers. So these are the persons who have to be careful in making smart decisions. Okay, we know that someone gets in love with someone that has a mutation. Well, love wins. There is nothing we can do. But they, okay, if possible, they should avoid having kids with a person that also has the mutation or is a carrier, because they may have children with severe conditions. In the case of autosomal dominant, before, we said that it usually manifests with or affecting structural proteins like collagen. Notice that autosomal recessive conditions manifest as enzymatic defects. Okay, one enzyme that is not made in enough amount, that doesn't work properly. And down here we have the probabilities, okay, the chances of passing a mutated allele to the offspring with each pregnancy. Okay, if both parents are carriers, they have 25% probabilities, 1 in 4, that the offspring inherits 2 copies and manifests the phenotype. Okay, if you go to the first slide here, is exactly this. Okay, in this case, it's not a disease. Blue eyes are not a disease, but in mind, we are talking about the mutation that produces a disease. 25%. And 1 in 2, 50% offspring will inherit the copy and will be carriers. Okay, are these 2 here? Okay, 25 plus 25%, this is 50%. And 1 in 4, 25%. Of the offspring in each pregnancy. That is important. Okay, sometimes these concepts confuse some students, because they think, well, if there is a 25% of these, there is a 50% of these, and if someone has 4 kids, they are going to have 1 with the disease, 1 with no copies, and 2 who are carriers. No, they have the same probabilities in each pregnancy. Okay, so they have 25% or 1 in 4 probabilities of having someone, a kid, without any kind of mutation. Okay, it's not a carrier and doesn't have the disease, of course. So let's take a look at the pedigree here. Okay, here you can see what we were explaining before. Notice the family tree. Okay, this male and this female, these parents, are both carriers. So they don't manifest the disease. And they have 3 kids. 2 didn't inherit. Oh, sorry, no, they have 4 kids. Okay, they have 4 kids. 1 didn't inherit any allele. 1 inherited both alleles, so manifests the phenotype. And 2 are carriers. So exactly as we mentioned before, 25, 50, and 25. 1, 1, 1. Okay, now, if we receive this patient with a disease, for example, cystic fibrosis, And maybe we ask, do you have any family history of persons with chronic respiratory diseases, with recurrent pneumonias, and this and this and that? And no, I don't have any family history. Because these parents, if they don't know they are carriers, they are not gonna tell their kid, or they are not going to do anything to avoid marrying another person or having kids with another person that is also a carrier. So for this person, there may not be any family history. Now, what happens with this person? This person has kids with another that doesn't have any mutation. It's a smart decision because they are going to have 4 kids. Okay, and notice that all the kids are carriers because this parent has 2 alleles. So all their kids will inherit a mutated allele. Now, these kids, they know they have a family history and they are going to make decisions when they are going to have kids of marrying someone without the mutation. So they don't have a child with cystic fibrosis or any other autosomal recessive disease. Okay, so you have exactly the same here, exactly the same thing that we have in this pedigree, but now representing the male, the female, both are carriers. Okay, they have 1 affected daughter, could be a boy, 1 unaffected son, could be a girl, and 2 carriers. The next inheritance pattern is X-linked recessive. Okay, in this case, it's a recessive disease. Okay, the person needs to inherit 2 copies of the mutated allele. But now this is a condition, okay, that is not affecting the autosomes. It affects the X chromosome. Okay, if a female, or for a female to manifest the disease, she needs to inherit the 2 copies, okay, from each parent. The X chromosome from the father and 1 of the X chromosomes from the mother, the one that has the mutation. Okay, and for females, this will manifest exactly as any autosomal recessive disease. If instead of X-linked recessive, we were talking about X-linked dominant, now females would need only 1 allele, okay, for them to manifest the disease. Now for males is different. Remember, males have only 1 X chromosome. Okay, they don't have the good copy. Okay, men are hemizygous for the X chromosome. Okay, so if they inherit a single copy, okay, that they will manifest the disease. Typically, females in this case are carriers. Remember, I said before, these in females manifests exactly as autosomal recessive. Okay, now the probabilities for a carrier female with each pregnancy, okay, they have a 50% probability, okay, that her sons will inherit the disease, and 1 in 2, 50% too, that her daughters will be carriers. So notice that for males and females, there is a 50% chance of being carrier in the case of female daughters, and inherit the disease, manifest the disease in case of males, the sons. Okay, now affected males will transmit the mutation only to her daughters, to all of them, because males don't pass the X chromosome to boys, only to their daughters. Now these daughters will become carriers, okay, and they don't pass any X chromosome to the sons. These daughters will become carriers unless, okay, the mother also has the mutated allele. In that case, they may inherit the 2 copies and will be affected. Okay. So here we have the pedigree. Okay, here we have a woman, okay, that is a carrier, a man that's not carrier of any mutation, and they have 4 kids. There is that 2 of them didn't inherit any mutation, Okay, and here we have a female who is a carrier, inherited one of the X chromosomes from the mother. The other X chromosome is the good copy from the father. But here we have a man that inherited the X chromosome with the mutation from the mother. And since males are hemizygous, they don't have the good copy, they will manifest the disease. This guy marries or has kids with a woman that is not a carrier. And notice that the sons don't inherit the mutation, they only inherit the Y chromosome. And the daughters, they inherit a good X chromosome from the mother and the mutated X chromosome from the father. Okay, and in these cases, is also this inheritance pattern is also characterized by skipping generations. Okay, we have exactly the same here, but in this case, it's the same: carrier mother and unaffected father. It's exactly the same, but represented with the little persons. In the case of X-linked dominant, okay, the males will pass the chromosomes to daughters only, and females to sons and daughters. But in this case, the difference is that only one copy is required for expressing the phenotype, so males and females can be affected. The only difference is that males will be affected more severely because of having only one chromosome. Okay, there is no backup copy of the genes that we have in the X chromosome. There we have the pedigree of X-linked dominant. Notice that there is no generations are skipped. Okay, how do we differentiate these from the autosomal dominant? Let's take a look. Look at this is a female that passed the disease. Only one copy is necessary, remember, to a female and a male. Now, notice that from female, the mutation passes to both sexes, but from males, the mutation passes only to females. Okay, that is the difference between X-linked and autosomal dominant. Let's come here. You have a male passing to male, female, female. Okay, that is not seen in the X-linked dominant. Okay, the male passes only to female. Now we have to talk about a couple of other inheritance patterns. This mitochondrial inheritance is not Mendelian. Okay, this is different from the Mendel's laws. What we have to understand about the mitochondrial disease is that during fecundation of the oocyte, okay, when we form our first cell, that is the zygote, the sperm cell doesn't contribute with many mitochondria. So all the mitochondria that we have, we could say all the mitochondria, we inherit them from our mothers. So another name for this inheritance pattern is maternal inheritance. And what diseases the people manifest when they have problems with the mitochondria? Well, just think in the tissues that have a very high metabolic activity, brain, muscle. Okay, what is going to be the inheritance pattern? Well, only from females to everybody, to males and females. All the kids will have inherit the condition. Okay, if you have an affected mother, okay, all the kids will be affected. Okay, if you have an affected father, none of the kids are going to be affected. And these conditions have a variable expressivity. Okay, some people are going to manifest with mild symptoms, moderate or more severe symptoms. Now, to finalize today, we have to talk about what we call multifactorial inheritance. Okay, multifactorial inheritance is very important for human cases for clinical medicine. Not only human, I mind for veterinarians too. We are talking about clinical physiology. And this refers to the type of genetic inheritance where a particular disease or trait manifests or is produced by the interaction of multiple genes, not a single gene, together with the action of environmental factors or exposures, maybe at work, maybe at home, okay, maybe a poison, maybe something that interacts with the genetic composition. So we say that people have a genetic predisposition when they inherit several genes. That's why this is called sometimes polygenic inheritance, complex inheritance. And there are diseases, if you make a list of diseases that are multifactorial, that are mostly genetic, and there are conditions that are 100% environmental. Okay, Duchenne muscular dystrophy. Okay, when we say 100% genetic, there is nothing that we can blame in the environment or that we can do, okay, to avoid this disease from manifesting. Then we have other conditions that are very, very, very close to the 100% genetic when we lack the certain enzymes. Now, we can avoid them, or at least avoid the manifestations and complications by simply not giving certain foods. Like, for example, do not give dairy products that contain galactose. Okay, don't give anything that contains aspartame or anything like that that increases the amount of phenylalanine, etc., in the blood, because they can't process those amino acids or that sugar. And then we have diseases that are in between. Okay, they have the ones here: hemophilia, osteogenesis imperfecta, is more to the genetic part than to the environmental. But then we start having other conditions: pyloric stenosis, hip dislocation, peptic ulcer, diabetes, schizophrenia, tuberculosis, that are more environmental. There is tuberculosis, infectious disease, it's very, very close to the 100% environmental. Okay, but still, people have some degree of genetic predisposition, very small contribution of genetics in this case. Scurvy is 100% environmental. There is no genetic predisposition. Everyone will suffer exactly the same if they have a deficiency of vitamin C. Will manifest with scurvy, with bleeding gums, with many problems as a result of this a lack of this nutrient, vitamin C. So, exactly as a person's height, for example, okay, is determined by several genes and environmental factors like nutrition, health status. Okay, these conditions will manifest or will manifest exactly if the combination of genetic predisposition and exposures to environmental differences, environmental factors, are together. Okay, we can say that about heart disease, cardiovascular disease, high blood pressure, Alzheimer's, diabetes, cancers. Okay, there has to be a combination of multiple factors with lifestyle, exposures at work, or smoking, or different other things. And here we have a list of just some examples of primarily genetic diseases. Okay, we are not studying pathophysiology, but it's a good idea to have some examples and start getting familiar with some of the conditions that we are going to be studying in the future. Cystic fibrosis is produced by a single gene mutation in this gene. CFTR is a gene that codes for a chloride channel. Okay, affects respiratory, digestive tract, producing pneumonias, diarrhea, malabsorption. Huntington is a disease that is very sad, appears as a result of genetic mutation. It's a mutation that produces a repeat of a triplet. Okay, we are going to study that. Is a triplet: cytosine, adenine, guanine, that is repeated many times tens and tens and tens of times, CAG, 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 and leads to the production of a protein in excess, a protein that is called huntingtin, and that protein accumulates and destroys the neurons, leading to a dementia with some motor disorders. Sickle cell disease, mutation in the hemoglobin gene, producing this excessive hemolysis as a result of the sickling of the red blood cells. And here we have some primarily environmental disorders. The lung cancer that results from smoking because of the carcinogens in tobacco that damage the DNA, leading to formation of cancer, uncontrolled cell growth. Uncontrolled cell growth and division. Now, skin cancer that results from exposure to ultraviolet radiation, okay, is mostly environmental. Okay, there is a genetic predisposition. People who are have a very light skin that burn easily, but if they avoid, okay, this exposure for a long time to ultraviolet radiation at certain time, if they use sunscreen, etc., they will avoid this cancer. And lastly, silicosis, which is produced by inhalation of silica dust. People who work in mining, for example, quarrying industries, leading to inflammation, chronic inflammation, scarring. These people develop nodular lesions in the upper parts of the lungs. Okay, and it's entirely environmental and occupational. Okay, so this is, or this was the basics for genetic disorders. Okay, there you have some extras, okay, with how to interpret a pedigree, okay, the different symbols that are used. Okay, you have there many different, you only need these for now, the ones that appear here. And also you have there some slides about some important terminologies, genetic terminology or genomic terminology that you will need.